Okay. Hey, Sister Mayo. This is our video for lesson 2. So the question for number 1 is I'll just read it. The ABO blood group in humans assorts independently from rhesus blood group trait and the MN group blood group trait. The alleles A and B and I are possible for the ABO locus. 2 alleles are a dominant allele producing RH, and dominant allele is RH positive, and the recessive or RH negative has to be homozygous recessive. And then MN, or alleles M and N are codominant. Child born, or a child with blood types O, RH negative, and M is born to a woman who has blood types O, RH negative, and MN, and a man who has blood types A, RH positive, and M. Determine a possible child. So the child in I, in order to be RH minus, had to be little r. And then what was the phenotype? M? So it has to be homozygous dominant for M because MN has codominance. In order to get this, parents that, well, the mom says that the blood type's O, so she also has the little I. And little r, and she's hetero. And then, so in order for both of these little i's to come from the dad had to have a little I, even though he's type A. So we have, he's going to be heterozygous. He's RH positive, but he has to be heterozygous in order for this second little r to get here. So he's big R, little r, and then he's M, right? Yeah. Then he's type M, so he's going to be homozygous for M. So these are the ways that this child can have this genotype. The next, what proportion of children to a man with the genotype AB, big R, little r, M, N, and a woman who is A, O, big R, little r, N, N, will have the blood types B, RH negative, and MN. So I did the Punnett squares for these, and I got the percentages. So I'm just going to write the percentages up here. So if you do the Punnett square for A, B, and O, you have 1/2 is A, type A, 1/4 is type B, and 1/4 is type AB. Yeah, type AB. Okay, and then the ratio for RH positive is 3:4 to 1/4 negative, and then for okay, then it's 1 over M and 1/2 MM.
Sorry, MM or MN would be 1/2.
MN's, oh yeah, MN's 1/2.
M would be 1/4 along with N.
Oh, that's right. M, then 1/4 N, N. And then the genotype, or the phenotype was B, RH negative, and MN. So you take B, which is 1/4, times RH negative, which is 1/4, times MN. Yeah, times MN, which is 1/2, and that equals 1/32. Yeah. And then C, we did the same things. It's a little bit of a different ratio, but in order to get type A, RH positive, and M, the ratio was 1/4 times 3 over
3
4
over 4.
times 1/4. 1/4 times 3/4 times 1/4. And that equals 3 out of 64, I believe. Yeah. And then the last one, this guy says the kid's not his, and we're trying to prove that.
Yeah. It has blood type A positive and MS. That RH positive and N as a name, the students can do that. R,
R, oh, this could be anything. And then
just N. Yeah.
And the kid's got what?
The child has O, RH negative, the little r, little r, and then M, N. Yeah.
Okay. So the kid totally could be his because being type B doesn't necessarily make you homozygous for B. He could be type B and have a little I, and the mom could also have a little I. And then to get these down here, you could also have, and they could be Rh positive, but still carry the negative or the little r here. And then the NN, he gets the M from the father. He'd have to get the M from his mom. But this could be his kid. And I actually found the probability of that, and it's 1 out of 32. And it's his kid. Okay. So that's question 1. I passed the baton.
All right, so in this question, we're going to be working with chickens. And so 4 different types of these chickens, right? We got the walnut. We've got rose, pea, and single. Walnut is got the dominant R, and that was dominant P as well, I believe.
Yeah.
And then rose has the dominant R, and then recessive, I guess recessive on that one. And this would be recessive R's, and then a dominant P, and single would be all homozygous recessive. And so for A, it's asking how would we get the situation where we can get 1 walnut, 1 rose, 1 pea, and 1 single coned chicken. And so what we did was just, instead of bothering to figure out what the parents were right off the bat, we just stuck in what the possibilities for the offspring would be. So then we just stuck in like an R, P1 single. So then we just stuck it all in. So then from this, we know that there's a little p right here. There's also a big P right here. And then also you had to get a big R right here. And then a little r here. And then you definitely have to have a little r here as well to get both r's here. Same thing here. And this one, these two don't matter. And then as far as the p's go, this doesn't matter, but this one needs a little p here, here. And so then the parent would be on this side, the R, big R, little p, and then this parent would be little r, big P, little p. So that's A. And now for B, we're getting 3 rows and then one single. And so for the rows, I guess the single would actually go right here. For the rows, you just need one big R. And then that doesn't matter, the little p's. And so in order to get both the little r's here, right, you need a little r and a little r. And then you also need a little p. All of them are going to be little p, really.
Right.
And then you need a big R here and then a big R here to get the big R's across, which would end up making this one. But anyways, that would give you the 3 rows and then the one single column, which make the parents big R, little p, big R, little p, little p. So that's B. And then C, we get a 1 rows to 1 single ratio, in which the rows, right, to single. And so you just need 1 big R here, and then everything is one parent, big R, little p, little p, little r, little r, little p, little p. That's for C. And the last one was 3 walnuts and 1 rose. So there's your, those are your 3 walnuts.
Mm-hmm.
Then you need 1 rose. Yep. R, a little p. And so all of these are going to be big R. Need little p's here. This has to be little p, but then you need the big p's here, big P, big P. That would create your parents. Like that.
Mm-hmm.
So that would be number 2.
All right, and for number 3, talking about the horses and the 3 different colors that you can get. And we came out with that it would be incomplete dominance because the Palominos is a blend of the chestnut and the cream or the cremello. And so for the possible genotypes, it would be for chestnut, it would be big C subscript B with another homozygous big C brown, and then Palomino.
Those are 2 subscript B's, right?
Yeah.
Okay.
Yeah. And then Palomino is a subscript C subscript white for the mellow, which is your homozygous C subscript W. And that is how we came up with number 3.
Yeah, perfect.
Yeah, we just noticed a very heterozygous relationship with the Palominos. So
Right.
we just came up with that conclusion.
And Dylan had a good point that both of the alleles are big C because neither of them is recessive. That they are both are neither of them masks the other one. Okay, so then we're on 4, and it's just fill in the blank. So we just went through the notes and the slides to get these, and it just, so the enzyme encoded for AB, or for the A and B alleles, is called glycosyltransferase. If a person has type B blood, this enzyme adds the sugar galactose to the surface carbohydrates on the red blood cells. And then for B, overdominance or heterozygote advantage is a genetic phenomenon where heterozygotes have a reproductive advantage over the two homozygote classes. C was incomplete penetrance, is a genetic phenomenon in which an individual does not express the phenotype specified by their genotype. And the example was 6 fingers. Epistasis is a genetic phenomenon where the alleles of one gene mask the phenotypic effects of the alleles of a second gene. And then the disease phenylketonuria, that's the one that, or in humans, can be alleviated by modifying a person's diet. The 2-to-1 phenotype, this is F, the 2-to-1 phenotypic ratio of a monohybrid cross with the Manx trait suggests a lethal allele inheritance. And then G, this is talking about the shell coiling of the snails. And so we basically just figured out that the genotype of the males is totally irrelevant in the phenotypes of the offspring. And so what we did was we realized that 100% of the homozygous dominant females, or the homozygous dominant females, would have 100% dextral offspring, whereas the offspring of the heterozygous individuals, or the females, would have 50% dextral, 50% sinistral. And then the homozygous recessive females will have 100% sinistral offspring. So to kind of just give like ratios, like to make it a little bit easier to understand with the numbers, we just said that the population had 100 females, so that made all of the percentages individuals. So if there, this was the ratio for the females. So if we said if each mother has, so if there's 30 moms here, 30 moms here, and 40 here, if each mother had 2 kids, that would result in 60 dextral from the homozygous dominant. There's a 50-50 chance that the offspring from the heterozygous mothers will be dextral or sinistral. So that results in a 30 dextral and 30 sinistral. And then if the homozygous recessive mothers have 2 kids, that results in 80 sinistral. So that doubled the population. So now it's out of 200. So we have 90 over 200 is our dextral. And that makes, what does that make it? 110? 110 out of 200 are sinistral. And then if you like just divide those, I mean, that ratio ends up being like a 9-to-11 ratio, or 45% and 55% dextral versus sinistral. And then the last one was about the 4 o'clock plants. And again, this one, so this inheritance was the maternal inheritance. Well, it's kind of the same for the 4 o'clock plants, except for it's extranuclear. They don't, I mean, it doesn't have to do with, has to do with the chloroplast being given to the offspring. And since none of the sperm or the pollen have chloroplasts because they're so small, all the chloroplasts come from the mom. So we can again disregard the phenotypic ratio of the men or of the males and just focus on the females. And since there's no combination of genes, if a green plant is going to pass on its chloroplasts to its offspring, so all of the green females are going to give green chloroplasts, all the white females are going to give white. Since there's none of the variegated leaves in this situation, we just kept the same ratio. So it's a 2 to 3 ratio, or 40% and 60%. And that is that.
All right, so moving on to number 5. We're working with guinea pigs now, kind of like the coat color with the rabbits in our little slideshow that we had. In this one, we're working with black coat color, which is more dominant than the sepia color, and then we have cream, and then we have white. But it could have just put C for coat color, but and so for A, looking at so we're determining the genotypes and phenotypes of parents that would produce the following offspring. So in A, we're given we want a 50% black, and we want a 50% sepia offspring. And so with this, we determined that you would need the dominant B to transfer for the black ones, but then you want the rest of them to be the recessive sepia color. And then this one doesn't really matter because this sepia would mask anything else that would be here. So it could be BC, BW, doesn't really matter. That would give you those offspring. So you have a black and then a sepia parents. And then for B, we're working with a 75% sepia offspring and 25% cream offspring. And so for this one, you're going to need a B with the S, the PC for the cream mixed with a S and also a BC. And because these would act as basically dominant ones, we can kind of get the same 1/4, 2/4, 1/4 Punnett square with this, but those are both, all 3 of those in the top would be black or sepia. Thank you. And for C, we would have we want 50% black. We want 25% sepia, and then we want 25% white offspring. And so for this, we want the dominant B to get our black ones, and then our BW right there to get our white eventually, but then BS and another BW. And so this would instantly give you the 50% because it would mask whatever's combined with it, both the BS and the BW. But then this BW would mix with this BS, creating your sepia, and then the BW would create your white on your Punnett square. And our last one, we're trying to make a 75% black offspring along with a 25% white offspring. And in this case, you're going to have your most dominant black one right there with a recessive white, and then the same thing again. So that way, the B carries down to both of these, making 2 blacks, and then this one would cover, I guess eventually the 2 whites would get together, making 25% white. And that is everything. Thanks.